So we've talked now about traits which are controlled by different types of genes, dominant genes or recessive genes. Talked about the words phenotype and genotype. And the activities we've done so far, like, for example, the dogs, the mouse genetics that you just did, some of our practice problems, they were all using relatively simple examples where our traits either dominant or recessive, and each trait is only specified by one set of alleles. It's either a black fur or white fur. It's either floppy ears or straight ears. Now, are most traits actually like that? They come in either, if you think about human characteristics, for example, do most human characteristics have like just an either or version? They don't. Like, do we have genes for our height? Like, is your height genetic? What do you think? Why are you saying this, JT?
Because like some people can be like way taller than their parents are, some people can be like way shorter than their parents are.
Okay. What about what's the tendency? Like if you have tall parents,
normally you're going to be tall.
Yeah, then there's probably a good likelihood you are also going to be tall. But it's not just an either or sort of thing. There's other factors. In fact, there's not a single allele that controls your height. There's not one single place on a chromosome that says, okay, this is exactly how tall you're going to be. Height is a variable trait. Now, there might be 5 or 6 or 10 different genes within our DNA, and they're all sort of influencing our height or other factors. Is anything else important besides genes for your height? Like, is there anything else that influences what your ultimate height's going to be?
Your posture?
Well, somewhat, maybe. I think it's something else.
Your like physical like health or whatever.
Yeah, things like nutrition, your physical health. So you may have genes that you inherited from your parents that are going to give you sort of a certain height. But if you have maybe poor nutrition, as a child, as you're growing, if you have high levels of stress or health problems, then that might affect your ultimate height that you're going to grow to. What we call that is your environment. Your environment interplays with your genes that eventually determines your final height. Think about something like skin color. Do we have genes that dictate our skin color? Yeah, right. We can inherit genes from our parents that determine how much melanin our cells in our skin produce. But does the environment also come into play? Do our cells ever produce more melanin? Yeah, they can. Okay, as you're exposed to more sunlight, you may produce more melanin and your skin become darker. So there's the environment, outside influences also affecting how our genes work. So we simplify things a lot when we're talking about genetics in this regard. Like the things we talked about today, there's almost no human traits that I could use as an example to you that actually follow this simplistic dominant or recessive inheritance pattern. It's much more complex than that. And so we simplify things to help us kind of understand. And there are some traits that are like this, especially like the plants Mendel was looking at. Many of the traits he examined, they really were either purple flowers or white flowers. But when we start to think about other examples, it starts to become more complex. So we're going to talk about some of that complexity here. There can be more than just 2 alleles for a certain trait, right? The alleles you were looking at for mouse fur color was either there was a black allele or a white allele. But sometimes there's multiple alleles. An example of that is fur color in these rabbits. So these are some rabbits. They're all the same species. Okay, these different domestic rabbits, and they could have a whole variety of different fur colors. In fact, there's 4 different alleles that determine the fur color of these rabbits. It's not just a black or white. There's a brown gene, a brown allele, a gray, there's a spotted one. And so the interplay of how these various genes are inherited determines what the rabbit's going to look like. Okay? And some are more dominant than others, but it's much more complex. Another example is human blood type. What can your blood type be? Like, what are the possibilities? Anybody know their blood type? I'm B, type B.
O negative.
You're O negative. Okay. So there's B, I'm B, you're O. What are the other versions that you could be?
I think I'm like A positive.
Could be A. We're going to ignore the positive negative for now.
AB.
Yeah. So that's the 4 basic blood types. There's A, B, O, and AB. There's 4 different blood types in those categories. And the reason for that is because there's 3 separate alleles for blood type. There's an A allele, there's a B allele, and then there's an O allele. And depending on which combination of those you inherit from your parents, that determines your blood type. Okay? If you get 2 of the A alleles, then you're type A blood. If you get 2 of the Bs, you're type B. If you get an A and a B, you're type AB blood. And then there's the type O allele. This is a recessive allele. So the only way you actually get type O blood, like any recessive trait, is you inherit 2 of those recessive alleles from each parent. And so you could see an example of a Punnett square here. Here you have a parent that has an A allele and a B allele, and another parent that has 2 of the O alleles. When we do our Punnett square, we end up with this: half of the children would have type A blood and half would have type B. None would have type O blood. None would have AB blood. And so inheritance of these traits can be more complicated than we typically talk about. There's also something called sex linkage. We talked about the sex chromosomes, which can be X and or Y chromosomes in most people. And we know that typically XY sex chromosomes result in someone that's biologically male and XX is female. But what do you remember about those chromosomes? The X versus the Y chromosome. When you were doing the karyotyping activity, what was the Y chromosome like? Yeah, it was a tiny little, almost like a fragment of a chromosome. The Y chromosome carries on it very little actual genetic information. Okay. And so what this means is any trait that's on the X chromosome, okay, is not on the Y chromosome. And so when you have these alleles that are carried on the X chromosome, it's if it's a recessive trait, it's easier for males to get it than females, because males have one X chromosome and one Y chromosome typically. And so if you only have one X, you only need to inherit one recessive allele to show the trait. So one example of this is like color blindness. Okay? Males are more likely to be colorblind. And that's because color blindness is often caused by a recessive allele that's found on the X chromosome. And so if a person has 2 X chromosomes, it's less likely for them to inherit 2 of these recessive genes. But if a person only has 1 X chromosome, they only need to inherit 1 recessive gene to have color blindness. And so we could see that here. Oftentimes when we're talking about these sex-linked traits, what we do is we include the sex chromosomes in the Punnett square like this. And then above them, we sort of write the genes. So for example, here is the father, has an X chromosome, a Y chromosome, and they have a normal vision allele. Okay. And here's the mother. She is heterozygous. She has a normal vision allele and an allele for color blindness. So when we do the Punnett square, what we find, okay, this person has 2 X chromosomes, and they would get the normal allele from each parent. So they would have normal vision. Here's another female. They got a normal gene from the father, and then they got the colorblind gene from the mother, but that's recessive. So they're actually not colorblind, but they could be a carrier, which means they carry the gene for colorblindness, even though they're not exhibiting that phenotype. If we look on this side, this is a son, got the X chromosome from the mother with the normal gene and a Y chromosome from the father. So this is a son that has normal vision. And then if we look at this last box, they got a Y chromosome from the father, and they got the colorblind gene from the mother. This would be a son that's colorblind. So sex-linked traits such as this are more likely to show up people that have an X and a Y chromosome. Hemophilia is another one. You may have talked about that in like Global or in AP Euro about hemophilia is a blood disease that affected the royal families of Europe. And because they intermarried between families often, then these recessive diseases became more prominent, more common. And hemophilia affected some of those royal families. And it was a sex-linked trait. So male heirs to the throne could have hemophilia, a blood disease in which they bleed excessively. So we often will try, especially if we're thinking about sometimes diseases that run in families, inherited genetic diseases. Often we might want to look at sort of a family history of who in this family has had this trait, this characteristic, this disease, and how is it being inherited? What's the pattern we can see? And we use a pedigree diagram to do this. A pedigree diagram basically shows the phenotype and sometimes the genotype of several generations of a family. And so typically it focuses on a single trait you're studying. And it can help you understand, is this trait dominant or recessive? Is it sex-linked? Who might be at risk of inheriting a certain disease and so forth. So we have some symbols that we use to create these sorts of diagrams. Okay. So typically what we use is squares representing males and circles for females. When people have offspring together, when they mate, they get connected side to side, and then their children go beneath them. So this pedigree shows 3 generations of people. Here are some people, here are their parents, and here are their grandparents. And we color in we shade in the symbols if they have whatever trait we're studying. You know, if this is a pedigree for color blindness, the people that are shaded in are colorblind. Okay? And so we can show those relationships using one of these diagrams. And we'll do a practice pedigree diagram in a minute. Now, we know that we inherit alleles and genes and chromosomes from our parents, and that leads to variation in offspring, right? You're different from your siblings because you inherited a different combination of chromosomes from each of your parents. And we always get half from each parent, right? We always get 23 chromosomes from mother and 23 from the father. But which of those chromosomes you get might determine things like your appearance. So you might look similar to a sibling, or you might not. So the random mixing of chromosomes during sexual reproduction results in variation, differences. But there's another factor that results in variation, and that is mutations. So we've talked about in both mitosis and meiosis, before the cell divides, the DNA needs to be copied. And an exact copy of the DNA is created. But it's not always exact. Sometimes when the chromosomes are replicating, a mistake happens. Something gets changed in the DNA, and we call that a mutation. It's just a random change in the DNA as it's being copied. And so if the DNA changes, the characteristics can change. Mutations happen all the time. Often they don't have any actual effect on the phenotype. Sometimes they might create a helpful variation. Sometimes it might be a harmful variation. Just sort of depends. It's just a random change. And so there's two main types of mutations. Chromosome alterations is a change in the number of chromosomes, like we talked about, Down syndrome or Klinefelter syndrome or Turner syndrome, things you saw when you did that karyotype activity. And if we are changing a chromosome, adding a whole extra chromosome, or if a whole chromosome ends up missing or swapped with another different pair of chromosomes, it can create significant effects on the individual, right? Because one chromosome carries the genes for many, many characteristics. So if you're missing a whole chromosome, that can have serious effects, okay? And so sometimes, like we know, Down syndrome, for example, the most common chromosome difference is an extra 21st chromosome. As we talked about, sometimes during meiosis, when the two pairs of chromosomes separate, sometimes they don't. Sometimes both of those chromosomes go to one side or the other. And that can lead to a sperm cell or an egg cell with an extra chromosome in it. And if that sperm or egg cell is one that's used in fertilization, the embryo might have that extra chromosome. And having this extra 21st chromosome results in all of the differences that we think of, that we associate with what we call Down syndrome, okay? Differences in digestive system, differences in development, things like that. Those are a result of having this extra chromosome. And as the age of the parents increases, the likelihood of those chromosomes not separating properly also increases. So older parents have a higher likelihood of having a child with a chromosome difference. There's others, though. Down syndrome is one, but there's others. Sometimes people are born with an extra 13th chromosome. It's called trisomy 13. Sometimes a person can be born with two X chromosomes and a Y chromosome. You saw that in the karyotype activity. That's called Klinefelter syndrome. Turner syndrome, a person's born with only one X chromosome. So there are variations, things that happen during meiosis that can lead to some of these differences. There are times when a whole extra set of chromosomes can be passed on. Doesn't happen in humans. Humans having a whole extra set of chromosomes results in an offspring that just doesn't develop. But it does happen in plants oftentimes. In fact, seedless watermelons have a whole extra set of chromosomes. And it's why there's no seeds, and it's why you can't grow a seedless watermelon from a seed. They're created through selective breeding. So there are instances when a whole extra set of chromosome can happen in the plant. All right, we'll talk about, I guess, let's go through gene mutations. So a change in the number of chromosomes or something like that is a chromosome mutation. But there's also these other mutations. When the DNA, as I was just talking about, when the DNA gets copied, sometimes something gets changed. Sometimes a letter of the DNA alphabet gets switched to something else. We're going to talk about DNA again in our next set of notes, but if you remember back, DNA is made of a series of nucleotides that have 4 possible bases, A, T, G, and C. That's what our DNA, our whole DNA code that makes us who we are is a series of these letters, A, T, G, and C. And when the DNA is being copied, if one of those letters gets switched, that results in what we call a mutation. These are much more common than those chromosome mutations that we just talked about. So often these little changes are not even noticeable in the phenotype, but sometimes they are. And sometimes they just happen randomly. Other times, environmental factors can cause them, like ultraviolet radiation can cause mutations in cells. And this is why exposure to ultraviolet radiations, like getting a sunburn, can damage your skin, can make increase a person's likelihood of developing something like skin cancer, because ultraviolet rays can damage the DNA, leading to cells becoming cancerous. Certain chemicals can do the same thing. And some examples, albinism. When a person has albinism, means their body produces no pigment, no melanin. So they have no pigment in their hair or their skin or their eyes. And this is caused by a change in one of these bases. One letter being switched can result in albinism. There's other disease. For example, cystic fibrosis is a lung disease. It's caused by one letter of the DNA being switched. And the result of that is the lungs produce mucus that's too thick. And people with cystic fibrosis often get lung infections and need to be carefully monitored. There's no cure for it right now. Right? If a person's born with that, they have that for their entire lifetime. I went to high school with a girl that had cystic fibrosis. She died in her early 20s from the disease. Since then, treatments have improved and a person's lifespan has increased, but there's still no cure. But as we're going to talk about, when we think about gene editing, that's sort of something, that's the sort of disease that scientists believe they could probably cure in the near future. If you could go into an embryo or a person and change this letter to the correct one, you can basically cure this disease. So it's something scientists are working on, is can they edit the genes in a person to cure diseases like cystic fibrosis or other things. Okay. And so the last is, it's not just the genes that determine the traits of an organism. Oftentimes the environment, outside, that we just mean outside factors, also influence traits. Here's an example of that. This is a rabbit. It's called the Himalayan rabbit. It has certain genes that give it its fur color. But if you strap an ice pack to it, the fur below that ice pack eventually grows in dark. The environment, in this case temperature, affects how the genes are actually expressed. Here are a bunch of plants that were grown in different types of light. So plants have genes that determine how much chlorophyll they produce. It makes them green. But other environmental factors can switch those on or off. When they're grown, for example, in darkness, that gene gets shut off and it's not expressed anymore. Twins, identical twins, have the exact same genes. Their DNA is exactly the same. But can they have differences in their appearance? They can. And so it's not just the gene. If the environment wasn't affecting development or their characteristics, they would look exactly identical, but that's not always the case. There are differences even between identical twins. In fact, one of the ways that scientists study what is genetic and what is environment is by looking at twins that were raised in different households. So is your preference for broccoli genetic? Is it in your genes that you like broccoli or don't like broccoli, or is it not? We talked about this before. Well, one way scientists study things like that is by looking at these identical twins. What they do is they have identical twins that were adopted by different families. And what they look at is, okay, follow these twins and look at how are they similar, how are they different? Because if they're identical twins, they have the exact same genes. If these two twins that were raised separately in different households, if they both love broccoli, what does that probably tell us about broccoli? It's a genetic trait because they both if they both like the exact same music, maybe that's a clue. Well, maybe our taste in music is genetic because they have the same genes. Or what if they have one of them loves to draw and the other hates drawing? What does that tell us? It's what? That it's probably not genetic, right? Because they have the same genes. So when we look at the differences between identical twins, it can help us understand a little bit about what traits are or are not inherited and how that affects us. So last slide. In humans, almost none of our traits, I can't actually think of a single trait that's actually simple dominant recessive, inherited by one set of alleles. Okay? Most are more complex. Most are actually inherited by several genes interacting also with the environment. There are, however, diseases that humans are caused by certain mutations, certain single allele differences. One of them is fetal ketonuria. This is caused by a recessive allele, and it's a disease that your body doesn't produce a certain enzyme that breaks down an amino acid. Sickle cell disease, again, it's caused by a single recessive gene that causes red blood cells to be misshapen. So there are certain diseases that are carried on these recessive traits, and often they are produced by